Hello, dear students. In last lecture, we have discussed about the solid impeachment devices for the sampling of air, uh, and we have discussed different types of air sampling devices with respect to solid impeachment devices. Now, in this lecture, we'll discuss about the liquid impeachment devices, and here we have to also discuss different types of impeachment devices with respect to liquid media. In case of solid media, solid impeachment devices we have used if already prepared solidified nuclear agar plate, and on that we have to directly incorporate the air so that the organisms which are present in the environment or air get trapped on the surface of the nutrient agar plate. Now, here before entering the air into the particular environment, we have to add media in the form of liquid, and after that we have to inoculate that particular media on a nutrient or petri plate, or you can say nutrient media plate. So in this liquid impingement devices, a specified volume of air is forced inside the liquid broth. It is nothing but the somewhat the mechanism or the requirement for the particular liquid or solid impingement device is somewhat similar. We have to add the particular environmental air forcefully inside the assembly which we have designed for the impeachment devices, and small opening or capillary tube and bubbler through the liquid. So here, before proceed further, we have to assemble our particular assembly with respect to the sampling, and we have to force the air through a liquid medium which is already sterile, and that air is forced by means of small opening capillary or bubble through the liquid. So the microorganisms which are present in the air get trapped in the broth, which then enumerated, or you can say count by portrait method. These different liquid impingement devices are nothing but the bead bubbler sampler, capillary impingement devices, and Lehman air sampler. So these are nothing but mostly used examples of liquid impingement devices. Here, different microorganisms we have forcefully entered into the capillary tube or the different assembly we have designed for the different devices. And we have to count the microbial growth by means of overbreed method. It is again one of the method for the isolation of pure culture. So let's start with the bead bubbler sampler. So in this, you can see that diagram, nothing but the bead bubbler sampler. In these devices, the air is bubble air bubbler is fitted in a sterile flask which is connected to the vacuum pump. So in the diagram we can see it is nothing but the flask which is already sterile, and in that we have to fitted by means of rubber cork a air bubbler through which we have forcefully enter the air so that it can get mixed into the nutrient media which is available at the bottom of that conical flask. The air is drawn in through the capillary of the bubbler and is forced into the nutrient broth, which contain sterile glass beads, which reduces the formation of foam, or which reduces the foam formation. So in this diagram, we can also see the air is forcefully entered through the this capillary, or we can say air bubbler, and here is the air is uh, entering into the critical flask, where the sterile beads and media are already we have over there, which is nothing but a nutrient broth and glass beads which are already sterile, so that the microorganisms which are present in the air get trapped into the nutrient media. The microorganisms are entrapped into the nutrient broth, and a small amount of broth. After that, the excess air is we have to remove by it. So another flow meter, or we can say we can apply here the vacuum pump, so that the excess air which is released from that broth and beads, we have to remove by means of vacuum pump. After the desired amount of air we have passed through that particular capillary, we have to take a small amount of sample from that nutrient broth, and we have to inoculate by means of whole blood method. Um, and we have to observe for the visible growth of the colonies. after 24-hour incubation, we can count or enumerate the microorganisms which are present on the particular air samples. So these are nothing but the basic mechanism and principle behind the Bead bubbler device by means of liquid impingement devices.